And also we are going to talk about like the advantage and applications of this platform. And then finally, I'll discuss about challenges and the solutions. And then I will conclude with the few remarks. So first, I will talk about the CRISPR-Cas9 platform in the genome editing. So the concept of genome editing has started in 1994, based on the work from Dr. Maria Jessin, where she introduced the double-strand break. We can introduce any part of the genome to create the target mutation in the genome by using the endonucleases. So she used the rare-cutting endonuclease to introduce the double-strand break in the genome. Through that, the double-strand break can be repaired by both NHEJ and also by the HRR mechanism to repair. So this is the first concept of using gene editing platform in the mammalian cells. So later, the application of this technology is limited because of the availability of the site, and also due to the very rare cutting frequency in the mammalian genome. To overcome this, in 2001, the universal endonucleases, like the fork one, was fused towards the DNA binding protein. So in 2001, they fused the endonuclease, like the fork one, with the zinc finger domain. As we see here, the zinc finger domain, it requires 3 base pair sequence to recognize the DNA strand, and then the fork one, when it trimerizes with the other strand, it cuts at the desired site. The other DNA fusion protein, DNA binding protein that fuses the endonuclease is talons. In compared to zinc finger nuclease, talons recognize 1 base pair sequence. So we have a lot of versatility in recognizing the sequence. So this is other the advancement happened in 2009 in the genome editing field. But if you look at both the zinc finger nuclease and then talons, it requires complex cloning and then complex mechanism to target the desired sequence. To overcome that, in 2012, there's a new platform came, is based on the CRISPR-Cas9, where unlike the zinc finger nuclease and talons, it uses RNA-guided endonuclease mechanism to clear the target sequence. So coming back to Cas9 and then the other remaining platform, the zinc finger nuclease, as I said before, it requires substantial amount of protein engineering, and then it can be easily delivered to the cells because of its smaller size. But talons, it also requires complex cloning, and then because of the larger size, it's very difficult to deliver inside the cells. And then, but it's highly specific. But unlike other two, zinc finger and then talon nuclease, the CRISPR-Cas9 platform can be easily delivered to the cells efficiently. And then it requires only the PAM motif to target the desired sequence. And then not only for genome editing, it can be used for the research purpose for identifying the gene functions. So the function of the CRISPR-Cas9 was first identified in the bacterial immune system. So when the bacteriophage infects the bacteria, the cells that are escape from the infections usually integrates the bacteriophage genome into the bacteriophage sequence into the bacterial genome in the 20 base pair signatures in the CRISPR array. So as you see here, when the bacteriophage infects, the 20 base pair sequence is incorporated into the bacterial genome in the CRISPR array sequence. So this 20 nucleotide sequence is after integration, it will be formed as a trans-RNA and then cRNA, and then later when the bacteriophage reinfects the bacterial cells, this can be used as the immune system to avoid the bacteriophage reinfection again. So based on this, this applications was used for the genome editing tool for the mammalian genome editing. So basically, unlike the bacterial component, they have made 2 different systems. One is the guide RNA sequence, that's the 20 base pair guide RNA sequence along with the Cas9 protein. So based on these 2 components, we can target any part of the genome, and then we can manipulate according towards the desired feedback. So coming back to that Cas9, like the Cas9 has two different lobes. One is the recognition lobe, like where it recognizes the DNA binding protein. Another one is the nucleus domain, so where it cuts the DNA strand. So if you look at the recognition motif, it has two different regions, the Rec1 and Rec2, and then the nucleus domain has two different regions. One is HNH, other one is RUVC. And then these two domains are integrated by the bridge helix. And then the other component that is needed for targeting the by Cas9, it requires guide RNA. So the guide RNA has the 3 stem blue structure along with the target strand that complement to that targeting regions. So by using this both guide RNA and then Cas9 protein, we can manipulate the genome at the desired locus. So after the CRISPR-Cas9 cut into the genome, there are two different endogenous DNA repair pathways occurs. One is through NHEJ pathway, and other one is through HDR pathway. In NHEJ, it leads to, after double strand break, it tries to repair by inserting the small insertion, and also by it causes small deletions. But in case of HDR, when we provide the donor DNA along with the desired sequence, it can be incorporated into the genome. So both the NHEJ and the HDR happens depending upon the cellular state. The NHEJ occurs both in the dividing and then in non-dividing cells. But in case of HDR, it occurs mainly in the dividing cells and especially at the G2 and S phase. So coming back to in detail about each NHEJ and HDR pathway. So after double strand break, if you want to disrupt the gene, like the NHEJ pathway can give rise to disruption of the gene by inserting the small insertions, and also it can cause small deletions. Both can lead to gene disruptions. And if the double strand break occurs in the gene sequence, like sometimes it can also give rise to inversions. But if it happens to two different chromosomes, it can also give rise to duplications and also deletion of the whole gene. But in case of HDR, when we provide after the double strand break, if we provide the donor template with the desired sequence, we can insert the desired gene into the desired locus, and also we can functionally replace the entire gene, and also we can delete the part of the gene and also the whole gene with the depending on the donor template availability. And in addition to that, we can also make a point mutation changes in the desired locus. So these are the main applications of Cas9 after making double strand break. So depending upon our requirement, we can alter for gene disruption or by inversion or by gene replacement. We can use the desired changes. So as I said, now the Cas9 nucleases are mainly used for knocking in the gene and also for the knockout of genes. So this is very useful when there's a disease-causing genes are present. We can knock out those genes, and when the gene is mutated, we can alter those sequences by using HDR-based methods. But in addition to the Cas9 nuclease, we also have, in addition to double strand break, we can also make nicking in the DNA, and then we can cause the desired base pair change. One is by using the base editing and also by the prime editing. And unlike double strand break and nickase, both the double strand break divide of Cas9 also used for the cellular functions. When the Cas9 is fused with the transcriptional activators and repressors, and also by the epigenetic modifiers, it can be used for the gene regulation purposes. So I will go through in detail of each function. So coming back to the Cas9-based nickase functions, there are two tools are mainly used. One is base editor, other one is the prime editor. The base editor has two different components. One is cytosine base editor. As you see here, the cytosine base editor uses nickases, the Cas9 nickases fused with the deaminase enzyme. So when the deaminase is present in the target region, it converts the target C present in the window into T. First, it deaminates cytosine to uracil, and later through cellular repair pathway, it converts into T. So if you want to convert any of the cytosine into T, we can use the cytosine base editors. in case of inosine base editor, is similar to that of cytosine base editor. Unlike cytosine base editor, it converts adenine to guanine. So as you see here, the target 8% of the editing window is converted into inosine, and then later through the cellular pathway, it converted into guanine. So these are the point mutations. If we want to get any point mutation and then correct any point mutations, we can use base editors. But the base editors are mainly applicable only for the conversion of purine to purines, like that for the transitions. The translation is not at all possible with the base editor. And to overcome that, like there's a recently developed platform called Prime Editor was used. So here the Cas9 nickase is fused with the reverse transcriptase enzyme. So when they fuse with the reverse transcriptase with the nickases, there are three types of possibilities can occur. One is like we can make all the transition and the translation mutations, and then also we can use for gene deletions, and then we can also incorporate gene up to 80 base pairs. So this is like a much more advanced version of the advanced version compared to the base editors. So next, like the dead Cas9 tools, unlike in the Cas9 nickase and Cas9, it does not cause any single strand break or double strand break in the genome. It uses dead Cas9 and fused with the effector domain. So when the effector domain is a transcriptional activator, it causes gene activation. And in case of if the effector domain is a repressor, it causes gene repressions. So basically, the Cas9 is guiding the guide RNA genome will be guided by the dead Cas9 with the effector domain and causes the gene activation and then repression. So just go through the just giving the overview of this approach. So in terms of gene activation, if the dead Cas9 is fused with the activators like P3 and it causes the activation of nearby genes. And in case of like dead Cas9 fused with the activators, it also results in the activation of the nearby genes. Other fusion is like the dead Cas9 fused the demethylase enzyme. It results in the demethylation of the methylated sequence in the genome and causes gene activation. And other application is the gene repression that can be accompanied by dead Cas9 fused with the repressors. So if the dead Cas9 is fused with LSD1, it causes histone demethylation, through that it causes downregulation of the gene expression. And in cases of KRAP, it causes a repression, and then the D-Cas9, the DNA methyltransferase, it causes methylation of the genome and then suppresses the gene expression. So depending upon our application, we can use different tools to causes the to prove the different disease patterns. So the advantage of CRISPR-Cas9 platform is a simple and easy tool compared to zinc finger nuclease and then the Cas9 platform, and it is cheaper because it does not require complex cloning and it requires only 2 components. It's easier to modify the genome with using this approach, and we usually achieve higher efficiency compared to the other 2 platforms. And then we can target multiple sequence at the similar time. So now I will go towards like each toolbox and the application detail for different disease models. First, I'll talk about NHJ-based application for the gene disruption. So there are many number of diseases are altered by using this toolbox, but I will talk about NHJ-based gene deletion for the HIV infections. So as everybody knows that the T cells expression of CXCR4 and the CXCR5 is important for HIV infection. So when we target the 2 receptors like the CXCR4 and the CCR5 with the guide RNA sequence and with the Cas9 protein, we can disturb the gene expression so that the T cells won't express those 2 receptors. Through that, we can overcome the HIV infection of in the T cells, so the patient has a survival advantage. So this is one of the approach for NHA-based approach for the gene disruption. In case of inversions in the hemophilia, like if you look at there are 2 in the hemophilia A, there are 2 types of inversion occurs. One is intron 1 inversion, and other one is intron 22 inversions. So because of the homology present in the near telomerase region and then with the factor 8, it causes inversion of the gene. So when we use CRISPR-Cas9 to cut the homology region, it can restore the normal gene function, both in intron 1 and then also intron 22 gene. And then the NHG-based approach can be also used for exon skipping. So this is mainly useful for muscular dystrophy. So where, like, we can design 2 different RNA to remove the defective exon, so that the exon will be removed from the sequence, and after that it will have the normal gene for its function. So this application is mainly used for the muscular dystrophy in the in vivo applications. So here, like, they mainly deleted the exon 22 of the dystrophin gene to regain its function. And then the next toolbox is like HTR, the CRISPR-Cas9 based HTR. So after double-strand break, if you provide ssRNA with the corrected gene, so in here what we are seeing is the sickle cell mutation. As we see here, like the point mutation in the beta-globin gene results in the sickling phenotype. So when we provide ssRNA with the normal gene function, and after double-strand break with ssRNA delivery, we can incorporate the correct gene. And then after incorporation of that, the corrected gene, it will have a normal beta-globin expression and it reverses the phenotype. So this is one of the example where we can use HTR-based approach for the gene correction. The next approach is like we can remove the defective gene and then we can put the normal function gene in the disease locus. For example, if you look at the VASP disease, like the VASP protein is normally defective in the different exons. So here the whole VASP gene is integrated into the start codon, so that the defective gene is not at all expressed, and then only the normal gene will be integrated at the start codon, so that we can see the reversal of the phenotype in this disease model. As you see here, through this approach, we can get up to 40% of disease correction in these patients. The next approach is the base editing tool. So as I said, there are 2 different base editors. One is cytosine base editor, other one is adenosine base editor. So the cytosine base editor is used for different applications, but here the conversion of cytosine to thymine, it causes 2 different applications. One is used for mainly for the terminating the oncogenes. Like if you look at the cancer, there are different type of mutation occurs that results in the activation of oncogenes. So through use of cytosine base editor, we can make single base pair changes like cytosine to thymine, which causes normal functional amino acids into the stop codon. To that, we can completely inhibit the production of the desired genes. So this approach can be efficiently used for the targeting the different cancers. The next tool is the ABE, like the adenosine base editor, where the adenosine base editor is used for the skipping of the exon. As I said before, like the muscular dystrophy, the one of the exon is defective. Like if you look at here, the 52 is the defective exon. Through the adenosine base editor, we can skip this exon, and then we can have a normal functional protein. So here, as we see here, the splice donor site is having G2 base pair. Through adenosine base pair, we can convert the adenosine to guanine, and through that, we can skip this exon, and then the patient will have the normal phenotype. And the latest tool is the prime editing tool. So as I said before, both the adenosine base editor and the cytosine base editor can be used for only the transitions, but not for the transversions. But through the prime editing, we can correct the mutation that involve the transversions. So one of the example is the sickle cell mutation, where we need to convert T into A, like in the transversion, like purine to pyrimidine. So pyrimidine to purine. Here, through the prime editing, like the sickle RBC can be converted into normal RBCs, and then we can have the functional beta globin after this. So this is the approach, like they are currently planning to use it for the treatment of sickle cell patients. So all this, like up to now, I was given like different toolbox and then application in different disease models. But these applications can be can be used in two different ways. One is by ex vivo, just extract the cells from the patients, and then we can genetically manipulate those cells for correction or for addition, and then we can reinfuse the patient to reverse the phenotype. In other way, we can have it in vivo, like we can use the gene delivery system with the CRISPR-Cas9 platform in the lentivirus or in the adenovirus, and then we infuse to the patient to have an in vivo genome editing. So in the coming slides, I'll be talking about example for both ex vivo and then in vivo editing. So first, I'll go through about ex vivo editing. For the ex vivo editing, one of the major advancement happened in the last year by correcting the sickle cell patients by using gene editing approaches. So basically, the sickle cell and the beta thal patients were mobilized and isolated the hematopoietic stem cells from them. And then these hematopoietic stem cells were genetically manipulated for either for gene that upregulates the gamma globin, which can compensate for the defective beta hemoglobin, or we can introduce some of the mutations that are beneficiary to introduce the gamma globin activations, and or we can correct the disease-causing mutations by using prime editing or through base editing. So currently there are 4 different approaches are used for beta neuropathies. The one that is currently in clinical trial are based on targeting the B7 enhancer. So last year, like the follow-up around like 1 year, it shows very good reversal of phenotype in the sickle cell patients. And the second approach is like the ex vivo editing is like for the cancer immunotherapy. So where the patient T cells are collected, and then they were genetically manipulated for T cell receptor, and then for the CAR engineering. So basically, the CAR constructs and the TCR receptors are mainly integrated to the endogenous locus, and then can be utilized for targeting the cancer cells. So mainly, the CAR cells are mainly targeting about CD19 antigen for B cell lymphoma and other tumors. Or some of the T cells that are isolated can be manipulated in the genome to remove the inhibitory factors so that we can expand those cells and utilize for the cancer therapy purpose. So basically, like there are 4 different strategies that are utilized for cancer therapy. So the T cells are collected, and then either like the T cell receptor can be added, and then CAR receptors also can be added to the desired locus, and the gene of interest like the PTN1 and other genes are isolated, are also targeted in the tumor also isolated in the T cell population to target against the tumor cells. Coming back to like the next one, so what I discussed up to now is the ex vivo based platform for the disease correction, and now like we move on to the in vivo based approaches. So one of the well-known in vivo based platform for disease correction is the retinal disorders. So where the AAV vector packed with the Cas9 or the lentivirus-like particle packed with the Cas9 with the desired guide RNA can be used for the disease correction. So here, like these are the different models and the different genes that are targeted with the retinal diseases. So basically, like the VEGF-A are like inactivated in some of the retinal diseases, and there are some genes are like modified or ablated by using the CRISPR-Cas9 platform. And in case of Merck and then PDEC, you can see that the target genes are incorporated into the cells. So this is one of the major application in vivo. The second major application is the muscular dystrophy, as I said before. The one of the exon in the muscular dystrophin is defective, like exon 52, that results in the like the skeletal muscle and then cardio muscle type does not produce any no dystrophin. So through base editing approach, we can, as I said before, we can incorporate exon skipping by using adenosine base editor. So the defective exon is skipped so that it can produce the functional dystrophin protein. As you see here, compared to the control mouse, we can see that the skeletal muscle express normal dystrophin gene. And also through the prime editing, we can insert the 2 base pair sequence, which can put again in the frame, and then we can restore the dystrophin functions. So these are the in vivo applications based on the prime editing and then base editing in the in vivo platform. And then the other approaches for the neurodegenerative disorders. So where like in the normal like brain, when there's a neuroinflammation and then neurodegeneration occurs, it results in activation of the different cytokines, and that leads to oxidative stress and the mitochondrial dysfunction, and then oxidative stress and then apoptosis that leads to different neurodegenerative diseases. So through the CRISPR-mediated platform, we can gene correct and then epigenetically modifying these genes so that we can improve the cognitive function and then memory enhancement of the brain. So based on these strategies, we can target the different neurodegenerative disorders based on the CRISPR-Cas9 platform. And coming back to hemophilia, so the CRISPR-Cas9 based are like mainly used for the preclinical approaches at the moment for correction of factor 8 and then factor 9 gene. So in the mouse model, the defective factor 9 can be incorporated in the defective factor 9 locus by using the HR-based method. And then we can see that the defective gene can be overcome by incorporating the active variants of factor 9. Similar approaches has been followed for factor 8, where the factor 8 was incorporated into the uniselect expressing the AAVS1 locus. So that we can have higher expression of factor 8. So these are 2 different strategies are followed for the CRISPR-Cas9 platform for the correction of hemophilia A and B. In addition to that, there are 3 different approaches are currently used. One more is using CRISPR-Cas9 platform for generating the anti-thrombin to have a sustainable editing for both hemophilia and A. And then in other approaches, the factor 8 was incorporated, factor 8 and then factor 9 was incorporated under the albumin locus in the hepatocytes, so that we can have the normal production of factor 8 from the liver. And as I said before, the NHJ based and then based approach can be used for the inversions, because there are most of the severe hemophilia patients have inversion 1 and inversion 22. This can be corrected by using the CRISPR-Cas9 platform. So these are the other 3 different approaches currently in preclinical platform for the treatment of hemophilia A at the moment. Based on what I presented now, these are the clinical trials that are ongoing at the moment for different disease models. That's varying from muscular dystrophy to beta hemopathies. So as we see here, it's mainly used for the immunotherapy and sickle cell disease and then thalassemia, and also for the cancer models. So if you look at the distribution of the gene editing platform, from 2009 up to 2019, we can see that the usage of Cas9 based platform for gene editing is gradually increasing because of its easy to use, and then the cost, and then the effective of manipulating genome at the higher efficiency. And this again can be delivered by different platforms. As I said before, it can be used by adenovirus, AAV, and then electroporation. Is mainly based on whether it's ex vivo or in vivo. And we can see that both the plasmid based and the adenovirus are commonly used for correcting these diseases, most of the diseases. In addition to treating the disease, the CRISPR-Cas9 platform also used for diagnostic purpose for the infection disease. So once we get the infection from the viruses and it comes to the human genome, this can be readily detected by using the Cas13 based system, once the Cas9 base 13 is activated, it can transactivate the probe, and that can be used for the diagnostic purpose. So these are commonly used now for the HPV infection, also even for the COVID. So now, like what I discussed up to now was the usage of these applications for therapeutic applications, but there are many challenges and then that need to be addressed for the future use. So one is the efficacy. So one is like the major issue is the editing efficiency and also for the targeting capacity. And then second one is the specificity. Because of the lot of off-target effects, and then the on-target, the bystander effects also give rise to a lot of challenges. And third one is the safety. Because of the Cas9 protein, like there is a lot of immunogenicity associated with that, and also it also leads to the potential oncogenesis. And there are other challenges in related to ethical concerns and regulatory challenges, and then also for the pre-GMP production associated cost. So first I'll talk about efficacy. So the efficacy is mainly associated with the Cas9 activity and the method of delivery, and also by the target DNA site. Since the Cas9 was evolved from the bacterial system, it's very difficult to target the mammalian cells, because mammalian cells like the nucleus are present. So we need to deliver the Cas9 protein inside the nucleus. So through different approaches, like the Cas9 was fused with the nuclear location sequences so that it can be readily delivered to the nucleus, and also it can be optimized for the codon optimized for the mammalian cell expression. Through that, the editing efficiency was further improved. And then the delivery options. Like the pDNA delivery is initially was used for the generating the ex vivo based platform, and the problem is like it leads to integration, and then it also causes a lot of off target because of the long persistence. So later, like the Cas9 mRNA came, and then it has the faster editing efficiency, but it does not cause any integration. But now, most of the Cas9-based approaches are used by RNP complex as a readily available Cas9 protein with the guide RNA. Because of the shorter editing efficiency, we get like reduced off-target effect on this method. But only the major challenge is like delivery of this complex inside the cells. The second thing is the editing efficiency, the target sites. If the target site is like present in the nucleosomes, or if it is methylated in the target regions, we get very low editing frequencies. So to overcome that, we have to design multiple guide RNAs near the targeting site to get the higher efficiency of the desired regions. So other limitation is the PAM requirement. Even like some of the regions of the genomes are limited with the PAM availability. To overcome that, like there are two different platforms initially developed. One is using by the different Cas orthologs, and also by the different variants of Cas9, which recognize different PAM motif, so that we can target almost different region of the genome with the almost different variants. So next we'll move on to the specificity. So as you see here, even that the guide RNA that target the desired sequence, even the single base variation in the target sequence result to a lot of target efforts. So when we are designing the guide RNA to target any region, this has to be considered before taking to the clinical trials. And other region is like to overcome the reduce off target, there are two different approaches can be used. One is by engineering the Cas9 with a high fidelity one. Like there are three different Cas9s are available. One is the HYPA Cas9, which has the like in high fidelity one, which has a reduced off target effect. And also we can also screen for a guide RNA with different modifications to reduce the off-target efforts, either by reducing the size and also like making the modification in the guide RNA, make them have a reduced off-target effects. In coming to on-target, like when you're using the NHTA-based approaches, we'll get multiple insertion deletions at the target locus. So we get the mixed phenotype. To overcome that, we like the base editor was used at the base pair conversions, but again, the base editors as like the bystander effects within the editing window. If there is multiple A's or C's are present, it converts into the other nucleotides. So this is one of the major problem associated with the on-target effect. To overcome this, like the prime editor was developed recently, which can be used for the design-based conversions. So in terms of safety, so this is the Cas9 is the bacterial protein. There's already a larger presence of pre-existing antibodies to Cas9 in most other people for the therapeutic applications. Like if you look at if you want to use Cas9 for in vivo applications, already a lot of Cas9 antibodies are present in the patient population, so that the use of Cas9 in the in vivo application will be limited. So this can be overcome by using the SVO-based approaches, and also we can use the genetically modified Cas9 epitopes, which has lower immunogenicity, can also be used for the therapeutic applications. And the second major issue with the Cas9-based approaches is like due to the p53 activation. So when Cas9-based approaches are used for the genome editing, it causes activation of the p53 pathway, so that the cell that are selected for is mainly based on the p53 inactive cells that are selected for the after genome editing. And also it leads to a lot of toxicity, like mainly due to the off-target efforts if it happened in the oncogenic site. So these tools need to be considered when we are using for the safety concerns. And then the other challenges that are associated with is the cost. So when we want to make these reagents up to the pre-GMP grade, it requires huge funding, and also the preclinical validation also required for the desired targets. And then delivery is a major challenge to get the effect, because when you're using as RFP complex, we have to use the electroporation and then other platform to have a higher editing efficiencies. And coming back to the ethical concerns, the gene editing is like already is a major concern using CRISPR-Cas9 platform. To overcome that, like the somatic approaches, somatic editing rather than gene editing can be used. And also there's a inequality in the treatment delivery because of the cost. And again, like if you look at the regulatory challenges, that's mainly related to IP, and then harmonizing the application towards international for the applications. So those are the main regulatory challenges we face when using this application in the clinical trials. Just to conclude, as I showed, the CRISPR is a highly effective tool, and it shows promising results in the preclinical studies, both in the beta-amyloidopathies and also in the muscular dystrophy models. And for several other diseases, is in the preclinical state. And there are different toolbox like nuclease, the Cas9 nuclease, base editors, prime editors can be used for different disease model depending on the mutation and depending on the function we want to achieve. Like if you want to do gene disturb and base modify, and if you want to do exon skipping, or if you do gene addition, these different toolbars can be used depending on the disease condition and also for the genome modification. Since like many of these applications are still in the preclinical stage, it will take other 5 to 10 years to achieve like the therapeutic state of use this generating platform for the therapeutic applications. But already, the last clinical trial from the sickle cell patients has shown promising results. So it gives hope that for other diseases also we can use it in the coming few years. And coming towards the ethical and then the safety concerns, it requires scientists and the clinicians and also the ethical people to work in healthcare and to ensure that the safe and effective, affordable outcomes for the treatment of different disease models. So I'd like to end my seminar with this. So I'd like to happy to take any questions.
Thanks so much, Mohan. We do have several questions that have come in, and I'll start off with this one, sir. And you kind of answered here in the last slide, but I'll ask it anyway. It says, when do we realistically anticipate gene editing for the bleeding disorders community specifically?
So as I said, like, the last year, there was a very successful thing, NMGA paper showed successful editing in the sickle cell and thalassemia patient. So this is a very good, shows hope for the other disease model corrections. So this is based on ex vivo platform, and they achieved like good editing rate and also reversal of phenotype even up to 1 year follow-up. And now this is like currently they are using for retinal disorders. They are planning to use and also for the immunotherapy. So I'm like, based on the previous studies, it's hopeful that like another 1 or 2 years, we can see that much more increase in the usage of this platform for the clinical applications.
Great. Thanks, Mohan. The next question that comes in, I understand that AAV gene therapy for hemophilia in clinical trials is not indicated for a pediatric population. What are your thoughts on gene editing technology for children?
So it's a great question. So one of the, we also did one big study in our institute looking at AAV prevalence in the pediatric population. We get 80 to 90% of pre-existing antibody in this population. So as I said here, so there are two different strategies can be used. One is ex vivo based approach, where we can ex vivo isolate hematopoietic stem cells. And then we are having like clinical trials for lentiviral based approach, where we can manipulate the hematopoietic stem cells ex vivo, and then we reinfuse to the patients so that we can reverse the hemophilia. So in that case, we don't have to worry about AAV based antibody. And then the second approach is like, even if we want to use AAV antibody, we can modulate the epitope of AAV in such a way that it can avoid the existing AAV antibodies. So these two approaches can be used. One is like ex vivo based, like translation of factor VIII into the hematopoietic stem cells, or modification of the AAV that we are currently using, AAV6 or AAV9.
Great. Thanks, Mohan. The next question asks, if the gene is edited, what are the implications for future generations, if any?
So up to now, like we are talking the perspective of disease correction. But as I said, there are a lot of ethical and then other concerns are associated, like if you want to use it for other applications. So up to now, like it shows promising results for the disease corrections. So like, like for the other main diseases, like other major disorders like neurodegeneration, and then for blindness, and then for cystic fibrosis, this can be potentially used. But for other applications, we need to work towards ethical concerns.
Great, thanks, Mohan. Next question that comes in your opinion, what do you think are the top 1 to 3 issues patients should know about gene editing for bleeding disorders?
This is a very nice question. Like, the major thing they have to look at about the cost, like, the only thing is like, as I say, like, is affordable, whether it's affordable to the patient. And the second thing is, what are the long-term consequences, what are the off-target regions that's going to cause the effect. And then the third thing is like whether it leads to any oncogenesis. So those are the 3 things they have to work towards. So the first reason is like work towards whether it has any potential off-target that leads to any long-term complications. And the second thing is about the P53 and other region, whether it can cause us any oncogenesis the later time. And third one is like whether that can be affordable for everybody in the disease populations. So those are the 3 things they have to look at.
Great, thanks, Mohan. And you touched on this question a little bit earlier, but I'll ask it. Maybe you would like to expand on it a little bit. But the question comes in: what are other rare diseases outside of bleeding disorders is gene editing being researched for? You mentioned some cancers and perhaps even heart disease, different things like that. Are there any other diseases outside the rare?
Yeah, so I think there are multiple disorders are used. Like I think if you look at the current clinical trials, like there are multiple disorders are treated at the moment. Mainly that anemia, sickle cell disease, and then thalassemia, and also like a muscular dystrophy, and then like ocular disorders and cystic fibrosis. So like variety of disorders are currently treated by using the gene editing platforms.
Great. And this question is along the same lines, maybe just to follow up on that. But what are the anticipated patient outcomes in those diseases where gene editing has been more utilized or researched, like cancer? Is it complete elimination of the disease? How successful has research been in accomplishing these?
Yeah, so we are looking for one-term solution, like unlike other approaches like shRNA and then like lentiviral based. We are thinking about one-term solution, like the disease is because of the defective gene. So basically, we are going to correct the disease gene with the genetic platform to correct, and then we expect the long-term cure. So basically, if you're using ex vivo based approach, we correct the disease phenotype in the hematopoietic stem cells or the stem cell of desired organ. We expect the lifelong cure. That's the major goal. Even as I said, the one study which was published in last year with the sickle cell disease and thalassemia, it shows like good follow-up, like with 1 year, the patients shows effective reversal of phenotype.
That's incredible. Thank you for elaborating on that. One of the next questions that comes in says, what will be the next steps to move gene editing into the bleeding disorders forward? So what are our next steps for that?
Yeah. So compared to other disorders, the bleeding disorders, is mainly at the preclinical stage, but especially in compared to other disorders, these are in the preclinical stage. But one group, like as shown, the factor VIII can be incorporated into the albumin locus, like in the University of Pennsylvania, UPenn. They are trying to incorporate factor VIII into the albumin locus. That's one of the promising approach currently is moving forward. I think they currently started the clinical trials, so we can see the progress in another 2 to 3 years.
That's great. That's incredible. And I'm sure you probably knew this question was going to come up. You talked a little bit about the ethics, but I'll ask you, what are some of the ethical concerns that you have heard from either the bleeding disorders or other communities regarding gene editing?
Yeah, so the bleeding disorders now is not yet started because in the preclinical stage. I'll come back towards the HIV-based approaches, like because you might have heard like there was a large ethical concern when the HIV, like the CCR5 gene, was edited in the babies without the proper approval, and we don't know what are the consequences associated with that. So we need proper regulation and then like IP and then ethical concern before doing any platform in the germline editing. In terms of somatic editing, we don't have to worry about much other than off-target efforts. But when we are going towards the germline editing, a lot of ethical and the IP-related things should be considered.
Great, thanks, Mohan. We have one more question that came in. Probably a good one to wrap it up here. But where can patients go to receive more information about gene editing and where the science currently stands?
So like probably when they're uploading the consent form, I think at the time of when they are receiving the treatment, or the healthcare professions, they can provide more information about the applications. Because there's a new technology coming up. So there'll be a lot of detailed applications will be coming in soon in the clinics and other places.
Great. Thanks, Juan. We really appreciate it. Appreciate your time. This has been fascinating. I know I speak for hopefully I can say I speak for everyone. A lot of the questions that came in were had a lot lot of comments and a lot of positive comments on there. But this information is fascinating. So thank you for sharing it with us, and thanks for taking the time to be here with us today. I'd like to, once again, thank you, Mohan, and thank everyone for joining us this afternoon. Please note that this recorded webinar will be available on Friday, February 4th at hemophilia.org under the events tab, with all of our other archived webinars. Also available in the events tab is our upcoming schedule for our weekly Wednesday webinar series. Thank you once again for joining us and presenting all this fascinating information. We appreciate you and your time, and have a wonderful afternoon.
Yeah, thanks, Brett, for giving this opportunity. Thank you.
Thank you.